A very good afternoon to all of you. Today the virtual lab that we are going to perform on the topic to study pollen germination through permanent slide. This one is the first experiment that you are going to do in your biology practical in class 12 syllabus. So see, there is two types of slide generally we use like one is permanent slide and another one is temporary slide. So this one is temporary slide that all of you are going to prepare. So what are the raw material and things required to perform this practical? Aim to study pollen germination through permanent slide. What are the materials that you need to perform this experiment? First of all, compound microscope on which we are going to observe our prepared temporary slide. Second, permanent slide of pollen grain on stigma available in the laboratory. This one is for the comparative study, and other raw materials that raw material also that we need in this experiment to perform, like first is your flower. Flowers should be mature. Okay, mature flowers, so that easily you can obtain the pollen grain from the anther. Second thing, you are going to need a glass slide. Okay, painting brush. Okay, painting brush 0 or 1 side. Okay, just to take the sample of the pollen grain so that we can easily dust that on the slide. Here one more thing that we need to prepare, that is nutrient medium. Okay, nutrient medium. See, germination of pollen tube from the pollen grain is not a simple process. Okay, it is going to need some special kind of nutrient. Cover slip and so on. Okay, two extra things we can need. So these are the main raw materials. See, there is a special group of chemicals which are we need to prepare the nutrient media that we will discuss in the further slide. Okay, or further topic. Of pollen tube. See, we all know very well, sexual reproduction is the process in which a new organism is developed by the fusion of male and female gamete. Okay, by the fusion of male and female gamete. So, what are the source of the male and female gamete? That also we need to know. Okay, so pollen grain is actually the source for the male gamete. Okay. Stamen is the male reproductive part of the flower that contains pollen grain. So where pollen grain is going to be present? In the anther or stamen. Okay. While carpel is the female reproductive organ of the flower that contains ovules. So ovule is the female reproductive part and stamen is your male reproductive part. Okay, stamen contains your pollen grain and your ovary. Okay, that is going to be, or carpel we can say, going to contain the ovule, which is the female gamete. Okay. Now what will happen? The pollen grain globular in shape with diameter. See, the shape of pollen grain is going to be vary. It generally range from 25 to 50 micrometer in diameter. Okay, the shape and size can be vary. It consists of two layers. Outer layer is known as exine, internal layer or inner layer is known as intine. After pollination of pollen grain, after pollination, if pollen grain reach on a compatible, this is what? Compatible. This is very much important thing, because it is not like if pollen grain fall on the stigma 100% there will be germination of pollen grain. No. Your stigma should be compatible, means should be in favorable conditions, so that it allows the germination of pollen tube from the pollen grain. Okay. It absorbs water and nutrients from stigma. Now, in time of the pollen grain emerge out through one of the germ pore present in the enzyme as pollen tube. See, when we are going to observe the structure of pollen grain, what will be happen? Suppose this is a general structure of pollen grain. Okay. Okay. So what you need to be considered here is that this region, okay, this region you can see that outer layer is not covering that particular region, okay. Exine and intine, this one outer one is your exine and inner one is your intine, okay. So you will see there is a special region where you are lacking the exine layer, which is actually made up of a special type of protein that is known as pollen. It is the most resistance molecules, biomolecules, okay. So through this region what will happen when a pollen grain fall on the stigma, compatible stigma, through these pores it is going to absorb the water and nutrient which allow the germination of pollen. So this region is known as germ pore. Germ pore. Got it? From there the pollen tube is going to be germinated. Come back to the topic. So this is the structure how pollen grain fall on the stigma, pollen tube germinate, and this pollen tube going to carry the two spore or two gametes. We can say one is vegetative and another one is generative. One is vegetative and another one is generative. Okay. So this is the initial stage. On completion of the process, you will see there this pollen tube grow, and there is a region that is known as chloral region. Okay. Chloral region or microphylla region in the structure of ovary. Okay, so this region, which one is having the filiform apparatus that is going to guide the pollen tube to make its entry inside the ovule. Okay, and through the pollen tube, these two gametes that you are seeing here, okay, mentioned here as a sperm, is going to make their entry. One of them going to be fused with the egg cell. Fused with the egg cell, that one is generative. Generative one is going to fuse with the egg cell, and vegetative one going to be fused with this polar central nucleus. Here you can see two nucleus are present. One more male gamete. That one steps between germination of pollen grain and formation of the seed that we will discuss in the theory. Now these include growth of pollen tube down the style. Style is the elongated region of the carpel. Down the style, release of two gametic nuclei, double internal fertilization. That's what you have studied, that in case of angiosperm, under the microscope, we are going to be observed by staining that with the cotton blue dye. Cotton blue dye, okay? What is the main reason, okay, to perform this experiment? To observe the various stages of the pollen germination through permanent spike. What are the various stages are there that we are going to be studying after completion of this particular experiment. So you can see the demo here. Okay, just have a look here how the mechanical